Hang on a second. Well, good evening, everybody, and welcome to the Fort Worth Astronomical Society's monthly meeting. My name is Bob Sirkis. I'm the president. I'm glad we have a pretty good sized crowd in the building tonight. How many, Phil? $24 plus me. I don't know if I count for one or not, but that's 25 people here. We're glad that you're here. We have a really outstanding program tonight, and Bill will talk to you about our presenter in a few minutes. So again, welcome everybody. The Fort Worth Astronomical Society has been in existence for over 70 years. And clearly we focus on astronomy. We have all kinds of interests. We have astrophotography. We're restarting our youth program. We have visual. We do tons and tons of outreach. Outreach is a big thing for me, and we work hard on that. Today, as of today, we have 195 members in the club. It's just outstanding. We have another month. We're trying to get 5 more to hit that magic 200. So when you hear me talk about magic 200, that's what I'm talking about. So if you know anybody that has an interest, please ask them to join us. It's a great club, a lot of interesting people, and we do a lot of very cool things. We have Allison as a visitor, and she's all excited I remembered her name. So, are there any other visitors here tonight? Can you? I'm sorry? New member. Okay, new members and visitors, excuse me. And I'm getting waved over here because I tend to wander and get off Chris's camera here. Can you introduce yourself? Awesome. Really cool. Sir? Okay. Well, our nickname, Sticky Fingers. Okay, so welcome. And maybe you'll see what we do and you'll want to participate. That's outstanding. Allison, what brings you here today? Okay. Awesome. Welcome. Hope you join. There's going to be a special deal for the 200th person that pays up. So anyway, welcome everybody. Welcome everybody that's online. And new member Karen Dole. Is she online? Oh. Anyway, so welcome Karen. Oh, well, welcome Karen Dole. So anyway, welcome. As we move along, ask questions, and yeah, let's get her started. Bill's going to introduce our presenter today.
You know all these people. Next slide. Do you? Why don't you mention the board members?
Okay. Okay. Yeah, Bill's I forgot to mention the board members, folks, and I apologize to the board. It's been a crazy day. Mr. Bill Radio Voice Hall is the vice president of the club. Monica Merritt is the treasurer, but I mean, she is. Just getting into town, so she won't be here today. And Mr. John Giormini, the most important guy in the group. He's our treasurer, and believe me, without the stuff that he does on the books and with membership, along with Phil Stage over here, we'd be in a world of hurt. And I know I'm forgetting somebody. So, so we have, oh, thank you. I'm with it. I was talking with a group of my teammates on the way over here. It was always exciting. So Phil Stage is director. He runs raffle. He helps manage the membership. And he is the history of the club. Robert Cargill is a director, and then Bruce Campbell has two very important roles. He's a director, and he is he takes care of making sure that Rising Star always looks like the 18th green at Augusta. He and his crew do an outstanding job. He's also on the development committee. They're doing some really outstanding work. Zach Smith is a director, and he is not here. I don't think he's online. And Cy Simonson, he's our legal beagle. He does a lot of stuff, especially in the last 6 months for us. And he's got some really exciting news, which at the end of the day, we're going to save money. So that's awesome. And then we have Patrick McMahon. Where are you, buddy? Patrick back here, he runs our events, our star parties. So when people log in and request events, it goes through him. And John is, can't see him, McCrae's back there. He takes care of, we have a special event that we do monthly at Tandy Hills, so he takes care of that for us. A lot of folks on the development committee, and they are in the process of planning the growth and what we're going to do out at Rising Star. Excuse me. Tom Roth is our Palo Pinto guy, and we're working to foster a partnership with the Thompson Observatory. It's in Keene, Texas. It's part of Southwest Adventist University. I don't know if George Lutch is here, but he is our webmaster. And as vice president, Bill Hall has the most important role of getting our meetings organized. And membership and donations is John Giormini. Next slide, please. And now, Mr. Radio Voice, Bill Hall.
Okay, so tonight we have Madhan Thurumalai, PhD. He's from the University of Houston. He's an astrobiologist, part of the Department of Biology and Biochemistry. A lot of his research involves not just biology on the Earth, but biology as it involves other planets, experiments on the ISS. He's been involved in research to explore the potential of how ascending probes to other planets could possibly pose a hazard as far as bringing Earth's biology to other planets. So I would love to present Mr. Madhan, or Dr. Madhan. All yours. I'm gonna turn the
Can you hear me now? Can you hear me? Can you hear me? Okay, good. So let me see if I can, I want to make sure that I put the right, okay, can you see my presentation? Hello?
Yes.
Oh, not yet? I think you need to make me host or make me co-host.
Let's see. Let's find out how. There we go. Okay, go ahead, try. Try to share your screen.
Okay. Can you see it now?
Not yet.
Interesting. Okay, I see some message, advanced sharing options. Okay, okay. I got it now. Okay, do you see my screen now?
Now we can.
Yeah, we can see it now.
Awesome. Okay. Let me put it as a slideshow. How about now?
Yes.
Okay, you don't see it as a slide, you actually see it full screen, right?
Yes.
Yes, the full screen.
Awesome. Okay. Hello, good evening, everybody. Thank you for the kind words, Bill. I really appreciate the introduction. So I did my PhD in India. It's been a long story. We can talk about it, my journey in the US later on. So I'll begin with a certain interesting slide, okay? The miracle is this: the more we share, the more we have. Can anybody here guess who said these words? Anybody on the chat? You can just unmute yourself and tell me. I give you 3 seconds. 1, 2, 3. You must all be familiar with this gentleman, Leonard Nimoy. These are his words. So I'm here to share something which I'm assuming most of you are not familiar with. In case you already know about me of the things that I'm about to share, so bear with me. It'll be like a reintroduction to whatever I'm about to share. So I'm going to talk about a field which is highly interdisciplinary astrobiology. This is the talk will be more like an overview of this field as well as the implications of some of the findings this field has for looking for life elsewhere. Okay, how many of you know this character, H.G. Wells? He wrote these books, right? The Time Machine, The Invisible Man, The Island of Dr. Moreau. The same H.G. Wells also wrote a book called The Crystal Egg, in which a shop owner by the name of Mr. Cave finds a crystal egg, and it serves as a window into the planet Mars. So as part of my outreach, oh, it's not moving. What happened? Okay, so I'm going to take a cue from Mr. Cave. If you had to use a time machine and come to the present, what would he discover? He would discover that the NASA Space Agency has an exclusive website for the field of astrobiology. To crudely put it, it defines it as a study that involves life here and beyond. Okay. Now it all began in the year 1984 when a piece of meteorite was, it's a fragment of a Martian meteorite. It's called Allen Hills 84001. It was discovered in Antarctica. And this sparked a lot of interest for the following reason. When they looked at it microscopically, they thought they observed fossils that look like structures of bacteria. But then, as it always happens in the scientific community, there'll always be a debate. People will start, literally punching at each other intellectually, sometimes even literally. And the whole idea of the hypothesis was rejected because the unusual features in the meteorite could well be explained by other processes that do not require life to be present. However, this eventually became a seminal event. It resulted in the emergence or the birth of the science of astrobiology. Okay, the discipline of astrobiology began around that time. So what does the discipline of astrobiology cover? It covers, number one, origins of life as we know it on planet Earth. Now the word life, I use it very carefully. We use it very carefully because it has both philosophical, theological connotations. So I would say, to be on the safe side, origins of biological life as we know it. And this involves the study of genomics and phylogenetic relationships between various organisms on planet Earth, life in extreme environments, which will be the main focus of this topic. And then we do something called laboratory prebiotic experiments, which is what I'm funded for in the last 1 year. And then there are some labs that also do complex organic simulations and observation. This is more towards modeling using computational methods. And the field also covers planetary systems, other planetary systems, planetary science in general. And then you have the Search for Extraterrestrial Intelligence Institute, which is in California, which constantly looks for signals from faraway galaxies for possible signs of life within quotes elsewhere. Last but not the least, it also interestingly includes the field of social sciences, humanities, and philosophy. Most importantly, education and outreach, because NASA is funded by the public, taxpayers' money. Therefore, outreach is an important component. It's an important component. However, the caveat is that not many people do this. I got involved in outreach since 2012. I can talk about it, how I got involved later, but since then, the bug bit me. I've been doing this kind of outreach events almost on a regular basis, with the exception of the COVID pandemic, which took away a lot of activities off the radar. So we'll begin with the extremophiles. Mr. K was present today. He would have discovered that his descendants, the two-legged creatures called humans, have actually discovered tons of extremophiles, which represent a world of mysteries by themselves. So there is one extremophile, which I'm assuming most of you know about. Therefore, I put the first slide. It's called the tardigrade. It's a close relative of insects and crustaceans. And it is also known as water bear. And it measures less than 0.02 inches in length, also known as moss piglets. And it can survive minus 270 degrees Celsius. From that extreme to something as high as 150 degrees Celsius. And it can also survive for decades at minus 20 degrees Celsius. It can also equally survive pressures from 0 atmosphere up to 1,200 atmosphere. So it is truly an extremophile, something that loves extremes, as well as radiation too. Okay. Sorry for the sound effect. That was actually made for an astronomy club meeting years ago, which also involves school kids, so bear with me. So if Mr. K was to see the present day Earth, he would have understood that an extreme environment is something that threatens access to basic requirements, the integrity of biomolecules like proteins, DNA, and RNA. And the environment also has limited resources for basic needs, and these are basically extremely stressful for life to exist. Here is an example for what happens in an extreme environment. When you make your cool omelette in the morning, you take the eggs and then you subject them to thermal denaturation. This is thermal denaturation at the bottom, and mechanical denaturation. What you do is you are actually denaturing the normal configuration of the proteins. Otherwise, they will be inedible. So this is an extreme condition to which you are subjecting the egg material to. So in life, we often come across substances from broad spectrum of extreme, for example, pH. Neutral pH is water at 7. If you go towards the alkaline pH side, you have from eggs to baking soda to all the way up to oven cleaners. And if you go towards acidic, from potatoes, bananas, tomatoes to vinegar, all the way to lemon and hydrochloric acid. Until the 1950s, most biologists wouldn't have believed that life could exist anything above or below 7. But life continues to surprise, biology, I'll correct myself, continues to surprise us by pushing the envelope of the extremities in which it can survive. So the word extremophile comes from two words, extremus, which is being on the outside, and philos means love, a love for extreme conditions. And the word was coined by Bob McElroy from NASA Ames. This is just to give you the background. So what are these extreme environments on our planet Earth? What are they and how do they look like? So these are some of the categorizations of extreme environments on Earth. Extremes of pH, as I explained earlier. Low pH means acidic, high pH means alkaline. Extreme salinity, very high salt, like the Great Salt Lake or the Dead Sea in Israel. Extremes of pressure, as in the ocean depths. Or extreme radiation. For example, the space radiation itself is extreme. The background radiation could be extreme. Or the radiation levels you find on Mars, about which I'll be talking. Extremes of temperature. It could be low temperature as you find in Antarctica. It could be high temperature as you would likely come across in a place like Yellowstone National Park. An extreme dryness or aridity. If you ever get a chance to go to the Death Valley National Park, you will understand what I mean. You have extreme dryness in the Death Valley, also extreme dryness in places like the Atacama Desert in South America. So let us begin with some of these conditions, okay, and see where they occur on Earth and what kind of organisms survive. So if you take a combination of low pH, that is acidic conditions, and high temperature, you just have to go several hundred miles up north in the Yellowstone National Park. And the Yellowstone National Park itself is a caldera. So what is a caldera? A caldera is like a loose piece of earth. It comes from the Spanish word, which means a cooking pot. Yellowstone National Park is literally a cooking pot, this piece of land that's underneath which you have like some boiling magma material. This is how this is a general depiction of a caldera. This is how the Yellowstone caldera looks. So what you see here is a granitic magma, and it's constantly pushing up. And as a result of which, you will have huge arrays of geysers and shallow hot water reservoirs. And if you've ever been to Yellowstone, there is this thing called Big Faithful that keeps erupting every now and then. So what is happening as a result of which is that it has created a unique environment of extremely hot and extremely acidic combination of heat and low pH conditions. And here is an example. This is a picture I took. So ignore the time on that because my camera was not set to the right time. This was the pre-iPhone days. What you can see here is the orange color flocculation. That's nothing but the sulfur that is getting precipitated out. And there is actually a sign here. I'm not sure if I have that picture here. This says, do not touch. The water is scalding hot. You don't want to touch even if it was not hot, because if it is not hot, it is going to be acidic. If you put your finger in, you'll probably lose it, either to the heat or the acidity or a combination of both. And here is the another picture. I think this is the Big Faithful. All these environments are characterized by high temperature and low pH. You see here? What you see here is the orange, yellowish sulfur along with the microbial mats. So when bacteria, microorganisms grow, both bacteria and archaea, archaea is a group of organisms classified under microbes. Over a period of time, they form something known as biofilms. How do you characterize biofilms? You get up in the morning, look in the bathroom mirror, stick your tongue out, you'll find a white layer, right? You can use a tongue cleaner, brush it off. That's nothing but a biofilm. The microbes have grown overnight, they have accumulated stuff, and they grow in communities like that. So when that happens over thousands or even millions of years of time, they form this nice mats of microbes. And what is incredible is that you can find these mats of microbes living at a very low pH, very high temperature. So that is an extreme condition. These are extremophiles. Yeah, I do have that picture. You see here, danger, hot water will scald. And these structures are made by a combination of minerals that get precipitated out from the hot water that comes out from the bottom, and the actions of the microbes. So mats of microbes that live in these hot runoffs, they form these terrace-like structures here. This is an incredible spot in the Yellowstone. For perspective, you can see the humans here. I'm not sure. Can you see my mouse moving? Are you able to see my mouse? Hello? Yes. Okay, good. Okay. You can see this is the structure that you see here, the terrace, they call it. It's not just the action of geochemical process. It's a combination of geochemical processes as well as microbial processes. Here is another close-up of the same. These are all made by a combination of both these biological as well as biological processes. And why are these important? Because when they look at things like this, and then they look at, they get pictures from Mars or any other exoplanet and other bodies, they look for structures like this that are unusual. And it is a speculation that scientists make. Could this have been formed because of some life either extant, still present, or extinct in the past? That's the general idea of why. And I mean, even if you're not looking for life here, so we would have still be exploring these, because the Earth itself, a majority of its ecosystems have been left unexplored in spite of 4 to 5 decades of research in this field. So when hot sulfurous water flows, it cools when it comes out. It results in precipitation of minerals, and it makes these little edges, and then microbes grow on them, forming biofilms, and then they get laid over with more and more mineral precipitation, and these terraces are formed. And thermophiles play a huge role in forming these structures. And what are thermophiles? So thermophiles, and let us look at the pH limits of various life. We looked at the pH limits in which daily substances are used. Fish can tolerate up to 4. So the idea is the lower the pH, the more acidic it is. There are insects like ephedrine flies that can survive up to 2. And then you have some plants that can go all the way up to go down all the way up to 2.5. But then you have microbes like algae that can survive 1, 2. That is as acidic as it can get. And then you have this one algae called Cynidium caldarium that can, the word caldarium, it comes from a caldera. It can survive 0 pH, which means that it is a pH that you don't want to go near. But life is surviving there, sometimes even thriving. And then you have these microbes like Thiobacillus and Sulfolobus, which can survive 0.2. So anything below 3 is super acidic. And we have abundant life thriving in those kind of environments. So here is another picture from Yellowstone. So each of these different layers of colors is a result of microbial growth, because there are different kinds of, because when the water comes out, it's not at the same temperature. Slight subtle differences in temperature results in a different community of microbes growing. So here are some examples of some of these Sulfolobus strains and their temperature and pH optima. Take the Sulfolobus, for example. It can survive all of them. Sulfolobus, Sulfolobales, Sulfobacillus, Acidocaldus. These are the different names, classification names, taxonomic names. You can see that they all survive between a pH optima. When I say pH optima, they require that low pH, acidic pH, and this very high temperature of 75 degrees Celsius. These are all in Celsius, not Fahrenheit. So a combination of these two conditions should be detrimental to life, biology as we knew it 50 years ago, but not anymore. Let's take a trip to Japan. So there's this place called Mount Yoti, where you see this yellow thing. This is nothing but sulfur. And it's meant to be detrimental to life. You can see a hiker, most likely a crazy scientist like me, trying to figure out if anything can survive here. And something does survive here. They found these two organisms called Picrophilus oshimi and Picrophilus torridus that do survive in these extreme conditions in Japan. And then let's take a trip to a place called Redding in California, which is characterized by acid mine drainage. So what happens is when you dig out, this is like a man-made extreme environment. When you dig up earth for mines and you leave it open, you have rains coming in, and a combination of these factors result in leaching, and that results in acidification. And what it does is, over a period of time, it becomes like an exclusive ecosystem for certain microbes to grow. Look at this picture now. Workers, according to the EPA, Environmental Protection Authority, workers once left a shovel standing in the green fluid flowing from one of the mine portals, okay? The next day, it just took 1 day's time, half of the shovel had been eaten away, not by the microbes, by the acidity of the drainage that was coming out. What is most astonishing is that in spite of the acidity, you have this, you see this red thing, it is nothing but a biofilm formed by microbial growth. Microbes thrive in this kind of a pH environment. You see here, this is an example of an acidophile, something that loves acid, Ferroplasma species biofilm. When I showed this to a high school gathering, they told me that this looks like feathers of a bird, like a peacock, but it's not. These are not feathers. These are actually these nice, long, undulating things that you see here. They are not feathers. They are actually biofilms formed by this particular organism. And what happened is, this grows at pH 0 to 0.5, super acidic, and 40 degrees Celsius, and it is attached to pyrite surfaces. These are iron ore surfaces. And what has happened is, over a period of time, the acid mine drainage being a man-made environment, this organism outcompeted all other organisms that might have existed there before. It became adapted and it evolved to survive in that, excluding, throwing out, pushing everything away. So it's a dominant organism there. So this was not known until 2000. So even today, as I speak, there are extremophile specialists continue to make such path-breaking discoveries. Yeah. So let's now go for a dive in the oceans. Okay. So the oceans are characterized by hydrothermal vents, which are nothing but undersea volcanoes, volcanic systems, or hot spring systems. Let's take a look at the overall morphology of the ocean floor. Look at the time. Okay, I'm on time. So if you go to the coastal side, usually it is, this is like a general morphology. You'll have the continental shelf, which is shallow. If you go to the west coast of India, where I did my PhD work, most of the shallow seas is characterized by 200 to 300 meters depth. And then you have the continental slope, shelf followed by continental slope, then you get the deep sea. The hydrothermal vents are usually found at the bottom here. As I said, these are underwater volcanoes, mini volcanic or hot spring systems underwater. And what do they have? They have, again, low pH, because think of the caldera that I spoke about earlier. This is a caldera at the bottom of the ocean, something similar to a caldera. And as a result of the water coming out, it's super hot. Suddenly it gets super chill. As a result of it, because of the water there, it's super cold. When hot water mixes with super cold water, it immediately precipitates most of the minerals. And these minerals form these nice chimneys and cool terrace-like structures in this hydrothermal vents. Of course, they are characterized by high pressure and most importantly, very high concentration of metals like cadmium, zinc, which are also thought to be detrimental to life or biology. But in those conditions, life manages to survive and thrive. So what are the, as I said, underwater volcanoes on the seafloor produce hot springs known as hydrothermal vents. And this is the ring of fire. Most of you must be familiar with this. So the ring of fire is characterized by these red dots throughout planet Earth. And wherever you have the ring of fire, you are likely to find the hydrothermal vents. One of the key hydrothermal vent system is in the northwestern region of the United States coast. It's called the UNT Heath Ridge, from where some real cool organisms have been isolated. In fact, the University of Washington and Washington State University in the state of Washington, they are pioneers in working in this area of research. Okay, what are some of the conditions? Number one, absence of light. So absence of sunlight, as a result of which they cannot perform photosynthesis. Therefore, chemical energy is utilized from sulfur compounds to fix carbon dioxide. And the superheated waters can reach up to 150 degrees Celsius. It's a conservative estimate. Sometimes you can even go up to 300 or 400 degrees Celsius. Extreme pressure and highly acidic, as I said earlier. Let's take a trip to one of these places called Tonga Islands. Keep an eye on the depth. It's 1,700 meters. That's close to what, 1 mile, just around a mile, right? And you have this champagne vents of Aifuku volcano at a depth of 1,607 meters. You're about to see a video of one of these champagne vents called the champagne vent. I hope you're able to hear the audio.
That's weird looking stuff. It's a really good picture.
You can see the pressure at which they are coming out.
Well, that was our assumption when we first started seeing the bubbles, was that it's some kind of a gas hydrate or clatrate that's decomposing.
Yeah. So I'll speak as you keep seeing this video.
Yeah,
There is life, abundant life around this place. It's extremes of fluctuating temperature. Yeah, you can see those rocks coming out. That's the pressure at which they are coming out. Yeah. So let's go to the UNT Heath coverage about which I spoke earlier. It is off the northwestern coast of the United States, the North American continent, off Seattle. What you see on the right is the geological morphology of this place with many ridges, oceanic ridges. And here is a black smoker called Sully. Probably the name Sully was derived by the person who discovered it. He probably had a pet dog or a cat by the name Sully. Therefore, he named it Sully. I do not know. I'm just guessing wildly. You can see that this black smoker, it emits jets of particle iron fluids. It is at 2,200 meters. From 1,600 now, we have come down to 2,200 meters. And what is interesting is this whole white and pink thing, they are actually tube worms. They are not microbes. They're actually slightly highly evolved organisms called tube worms. And they have been found. And when they were initially found, they were like, so they were taken aback. How can these guys survive? It turns out they also survive. One of the reasons they are able to survive is because they are assisted by a group of organisms known as symbionts. They live in close association with them, and these are microbes. Without these microbes, these guys will die. In other words, there's a close community relationship. It is like how the city of Conroe near Houston has a lake from which the city of Houston gets water. We have a close understanding. When the Houston faces water scarcity, hey, buddy, can you give us some water? Then it gives, and trade-off, we give them something back. Something, in a crude analogy, but you get the idea, right? So it's like a trade-off in evolutionary terms. But they are able to survive here mainly because of the extremophilic microbes with whom they are associated with. And the temperatures, as I said, 300 to 400 degrees Celsius. The pH is ridiculously low, 2 to 5. And these fluids, they transport very high concentrations of metals like iron, copper, and zinc, along with hydrogen sulfide. If you want to know about hydrogen sulfide, go to the Texas city, which is south of here, near Galveston. You can smell sulfur because they all have this oil reservoirs. Sometimes you get the smell of sulfur. So the black smoke is a result of fluid, such kind of fluid mixing with seawater. One particular bacterial strain, microbial strain has been isolated. It's called Geobacter. It's from a vent called Fin in the same ridge. It is roughly 1.5 miles deep, and it can survive between, this is a crude picture of this microbe. What you see, the strings here, they are actually structures called flagella with which it can move around. It can survive between 85 to 121 degrees Celsius. And let's go now to high pH, that is alkaline. We spoke about low pH and high temperature. We'll talk about high pH and hypersaline. For that, we move to Africa. If you ever go, this is Kilimanjaro. Most of you must be familiar. If you ever pay a visit to Africa, Africa is not only famous for the wildlife safari, it is also famous, not as famous, but it should be equally famous for something else. If you go to the border of Kenya and Tanzania, can anybody see the picture of the plane here? You see this red thing? This is unlike anything you have seen. Even in the Great Salt Lake in Utah, you won't see this. This is nothing but a whole big giant mat of microbes giving this red color. And this is for perspective, this is the shadow of the plane flying over it. So there's a place called Lake Natron, which is dominated by the red pigmented salt-loving algae. And these temperatures, they're not very high, but still for a conventional microbe, 50 degrees Celsius is pretty high. They survive in this 50 degrees Celsius. And pH, as opposed to the earlier organisms, is now on the other side of the neutral pH, 9 to 10.5, which is very, very alkaline. And this lake has an unusual mineral content leaked from the surrounding volcanoes. Once upon a time, if you watch the National Geographic videos of lion safaris, they'll talk about the Ngorongoro crater. The Ngorongoro crater was once upon a time volcano. Now it is extinct, but volcano means rich alluvial soil. lava soil, and that is the reason why this place gets unusual mineral content. Then we move to a place in California about which I spoke earlier. So this is called Owens Lake. This is also characterized by red pigmented halophiles. It need not just be algae, it could also be bacteria and archaea. And two things that characterize these organisms: they require large amounts of sodium chloride. Some of them have an absolute obligate requirement for have high salt. And what they do is to counter this, just for the biological phenomenon, how they manage to survive. In order to counter the high salt concentration outside, they accumulate high levels of solutes like potassium chloride to balance the osmotic pressure. Otherwise, the cells will burst. That is evolutionary adaptation. And they also express a pigment called halorhodopsin. This is the structure of the protein. It's nothing but a protein. And what it does is, in these high salt environment, there is less access to food. So sometimes they can also tap sunlight to generate energy. And then now, Mr. Keeve is wondering, what about Mars-like conditions? Because that's how we began talking about this, right? In order to do so, we need to know a little bit about Mars. So what does Mars have to offer? It lacks global magnetic field, very thin atmosphere, as a result of which you have a large amount of radiation that reaches the Martian surface. And you have salty brines that are found under the Mars surface. And it also contains perchlorate, which is usually found in places like the desert in Nevada. And it is the most Earth-like planet. And it is characterized by enormous amounts of ice. Okay, and that's one of the reasons why they're trying to see if Mars might have been inhabited by some kind of a microbial life in the past or in the future, or in the present, so not in the future. So in order to do this, astrobiologists have characterized certain places on planet Earth as different types of Mars analog environments. You can find them in the Canadian Arctic, the dry Mars in Atacama, in Antarctica, wet Mars in Rio Tinto in Spain, and then there's a wet Mars in Australia. These are all analogs for Mars-like environments. Yeah, let us begin with Antarctica. Antarctica, it's supposed to be like a super cold desert. However, so this is a picture of cool picture of one of the ice shelf. That's a piece of which is breaking out into the ocean. There's a deep lake in Antarctica called the Antarctic Salt Lake, which is 50 meters below sea level and never freezes. And not only that, it is home to these really, really cool microbes. So it is not devoid of life. That's the thumb rule. And here are some of the papers that talk about how life exists at 2.5 meters, the temperature that can survive a broad range of temperatures from 31 to 0.9 degrees Celsius. And then the average salinity of seawater is 3.5%. But here in these Antarctic brines or high salt reservoirs, the salt concentration is 18.8%, and life survives and thrives there. And there is one in Alaska where you have found sub-zero brines. This could also be called an analog for Martian brines, where they have found dense bacterial communities. And let's pay a trip to the Atacama Desert. So Atacama is an important dry Mars analog. And what you see on top here is a mosaic of images taken by the NASA Mars Curiosity. And this picture on the right here at the bottom is the Atacama Desert. You can see the stark similarity. And not only that, it has a recorded precipitation of very, very low single rainfall event of 2.3 millimeters, and one of the driest and oldest deserts on Earth. Okay, it has been super dry, arid for the last 90 million years. And you must all be familiar with this, right? This is Harvey, right? So Hurricane Harvey, it dumped 52 inches of rain over the Houston neighborhood in August 2018. Or is it 2017? Maybe I got the year wrong. It's probably 2017. Okay. If the same were to happen in Atacama, it would take at least 2,250 years to get the same cumulative rain in the Atacama Desert. That too, if it rains. It hardly rains there. So that's the comparison. Think of Harvey. It would have taken at least 2,200 years in Atacama to get the same amount of rain. It is that dry. Okay. So what are the conditions? Low oxygen because it is high altitude, very high UV radiation because it's cloudless, Desiccation means highly dry. And often you have winds and cold temperatures very similar to Mars. Mars is also characterized by high radiation. But do these conditions augur for life or biology? Yes, they have found fossilized stromatolites. What are stromatolites? Stromatolites are nothing but these biofilms I spoke about. Take the I gave you the example of the biofilm on your tongue. Imagine on a large scale, when they grow and get fossilized over millions of years, they form stromatolites. Okay, these are some of the examples of stromatolites found in the Atacama Desert. These have been created by microbial life. And this is one example where, lack of humidity, right? Life needs water, biology needs water. Where the microbial life here, it grabs moisture from the little bit of fog that comes from lower ranges, and that moisture it uses to grow. Okay, it's a fog that's coming in, it grabs these moisture. It has developed methods to grab the moisture, and they're still trying to figure out how they manage to do it, because it's not as if they have some kind of a drone to go and capture the moisture, right? They have cellular features that enable them to do so. So they're trying to figure out how they do it. So that is the life in Atacama Desert. That's one dry Mars analog. We'll take a leaf from there, and then we'll go to the Death Valley National Park. So Death Valley National Park, it's the hottest and driest place in North America. If you have never been there, I strongly recommend you pay a visit. Not during summer, it's going to be super hot. Probably during early winter or during spring. It receives about 1.5 inches of rainfall annually. One of the driest places, not as dry as Atacama, but still relatively dry compared to the rest of the North American continent. So why is it dry? Because it has these three mountain ranges, Sierra Nevada, Argus, and Panamint, which ensure that the rain-bearing clouds never ever reach there. That's the reason why it's a desert. And here, as it's called the rain shadow effect, as a result of which it is extremely dry. And it also resembles Mars because the overall topology of this region and also the surface resembles what one sees on Mars as well. Wind-blown sediments on Mars, the same thing you also see on in Death Valley. And if you go to a place called Furnace Creek, it is at 190 feet below sea level. That's another interesting geological feature. And it is so Mars-like that Hollywood decided to use this for making Star Wars. Star Wars. Sorry, I couldn't help it. I had to add that soundtrack. So RTD2, when it is making its way across the sand, this sand dune was actually in the Death Valley in Mars. Okay, and so it's a fun slide. Okay, so to keep people awake. And even in the Death Valley, they have found billion-year-old stromatolites. Stromatolites, as I spoke about earlier, these are microbial mats, biofilms that have been fossilized over 1 billion years. They've found in the Death Valley. These are the stromatolites fossils that are found in the Death Valley. And NASA also used this place as an analog for testing the Curiosity Mars in the sand dunes. So it's a fun slide. Anyway, now we move to another important extreme environment. This is again a man-made environment. So these are the spacecraft assembly facilities. So what you see here is a Jet Propulsion Laboratory in Caltech, California. So these environments are subject to very high levels of cleanliness. When I say if not super clean, if not sterile, super clean, okay? So they keep it dry, and they often subject many parts to extreme radiation. Why do they do so? Because there is something called planetary protection policy, and the planetary protection policy envisages, let's take the help of ET to explain to this, how can we protect other worlds from bacteria from Earth? When we send unmanned or manned missions to other planets, like when we send it to something like the moon, how do we make sure that we don't dump something from here there? There's nobody on moon to find us, but then we have to be more responsible. And in case there is some diabolical life elsewhere, how do we protect ourselves from them? This is what the NASA planetary protection means. It's not just about NASA alone. Every major space agency in the world has a protection policy. There's an international treaty for the same as well. So every space mission that is deployed has to follow certain rigorous procedures. So the whole planetary protection policy is to ensure minimizing forward contamination and totally avoiding backward contamination because we don't want to have the hypothetical Andromeda strain infecting us. And it's like that movie, Invasion of the Body Snatchers. We all become clones of plants. We don't want that, right? Yeah. So how do they ensure? In order to do this, they subject these spacecraft clean room facility where they are assembled to often radiation and sometimes even dry heat sterilization. Here, you can see that most of the technicians working during the assembly of the Phoenix Mars lander, they are dressed like surgeons. They don't look like engineers. They're dressed like surgeons working in operating theater because they have to make sure that the body microbes don't get transferred to this equipment. So a combination of keeping the environment nutrient poor, controlling the moisture and temperature, and extreme air filtering has created a special extreme habitat for bacteria and microbes that can resist all these conditions. So a clean room is actually an extreme habitat for microbes, not unlike what you find on Mars, because you also have the combination of radiation here. So as far back as in 1967, they found that these spacecraft also were carrying some of the microbes. This is as far back as in 1967. So this is definitely a huge concern. So in spite of all these conditions, as I said, a number of novel microbial species showing elevated resistance to desiccation, high dryness, peroxide, UV and gamma radiation have been isolated from these environments. Most predominant amongst them are these spore-forming bacillus species. So what are spores? Spores are nothing but inert forms of the bacteria. They can remain dormant like in this condition for millions of years. When they find the right conditions, they can grow. The fear or the concern is that these spores, if they can survive the Mars-like conditions in these clean room facilities, if they ride piggyback, they can contaminate other planets. We don't want to do that. So these are resistant to UV and gamma radiation. This is an example of the strain that I work with. I won't go deep into this because that will take another separate session. Here's the take-home message. The unusual radiation resistance of these contaminants found in these facilities, they seem to be suggesting that these facilities are facilitating a rapid evolution of these organisms. They have become like breeding grounds, and they may likely survive rigors of interplanetary transfer. We don't want that. That's why all these protocols are necessary to further reduce the bioburden by employing people like me to make sure we understand why they become resistant, etc. So let us now go forward and talk about this is the final part of my talk. We talk about high radiation, right? Radiation is extremely critical for deciding how life can survive or not, okay? In order to do so, I'm going to say a few words about how radiation affects biology, okay? Here is a spectrum of radiation, right? Short wavelengths, they result in ionizing radiation. What is ionizing radiation? For example, cosmic rays, gamma rays, X-rays, ultraviolet, and not ultraviolet. This is radiation that has enough energy. They can ionize molecules or remove electrons from atoms. Absolutely detrimental, okay? And then you have UV radiation, which is non-ionizing. It can rearrange a molecule structure, leading to DNA or protein damage. So neither is ionizing radiation good, nor is the UV radiation good, okay? And why? Because DNA and proteins, they are the main targets for cell damage from radiation. What you see here is a genome, and what you see is the protein. The gene codes for the protein, okay? That's the basic. You don't have to know more than that from this talk. So all you have to take home message is that the DNA and the proteins are the main targets of radiation damage. And in biology, if an organism suffers DNA repair, it tries to undergoes DNA damage, it tries to repair it. If the efficiency of DNA repair is on par with DNA damage, everything is cool, okay? However, if the DNA damage rate outcompetes DNA repair rate, then we have a problem because the cells will not be able to repair their DNA, and their survival rate will go down, okay? Vast majority of organisms on Earth are very, very radiation sensitive. Think Chernobyl, think Hiroshima, Nagasaki, think of any of the scares that comes from, what will happen in Russia, Ukraine. Maybe they'll use nukes. I mean, these are all fears, right? They are killed by doses less than 500 grays of radiation. So what is a gray of? It's gray is a unit, okay? What other organisms do we know that are super resistant to high radiation levels? These are all very important. They have implications for us to not only understand radiation resistance. Maybe we can exploit these organisms to protect ourselves. Ionizing radiation. You must all be familiar with this gentleman. He's a character from The Simpsons, right? 5 gray of radiation can kill a human. I want you to keep this in mind. 5 grays of radiation can kill a human. 200 to 800 grays are required to kill E. coli, which is a bacterium. Think about it. Who is more evolved, we or the bacterium? The bacterium seems to survive a much higher level of radiation than us. And there is this hyperthermophile, heat-loving organism called Pyrococcus. It can withstand up to 2,500 grays of radiation. Think 5 grays and 2,500 grays. Isn't that incredible? But that's not incredible enough. Wait for this. The best is yet to come. There is an organism called Deinococcus radiodurans, which was not radiation resistant, but we accidentally made it radiation resistant during experiments. It can withstand up to 5,000 grays of ionizing radiation. Think 5 and think 5,000. 5,000 times more. 1,000 times more, sorry. And then there is this organism called, it's a cyanobacterium, which are considered to be one of the oldest organisms. They are found all over planet Earth. They can survive a broad range of conditions. Okay, you will find them in hypersaline lakes, hot springs, high temperature, low temperature Antarctica, and arid deserts. Very versatile. It's like a musician who can play the guitar, who can play the violin, who can play the drums, who can sing soprano, who can sing tenor, who can also sing the blues and the Texan folk style guitar. It's like that. It's like an organism that can survive all kinds of conditions. This is how it looks. And it can tolerate up to 4,000 gray radiation. Okay. And here is an extreme halophile. I think this is from the Great Salt Lake. It can survive up to 5,000 grays of gamma radiation, ionizing radiation. And it now turns out that many of these organisms, their extreme nature are interlinked. If something is resistant to dryness, it will eventually become resistant to radiation and vice versa. And this has implications for Mars hypersaline subsurface lakes, because at least these guys have to survive both radiation as well as hypersaline. In the subsurface, they are shielded from the radiation. So the question is, can Mars hypersaline subsurface have potentially harbor these organisms? So these organisms are perfect analogs for potential Mars-like organisms. And then you have this organism called Rubrobacter. Look at this, it's 11,000 grays. 5 grays can kill a human being. If this guy can survive 11,000 gray of radiation, as if this is not enough, the mother of them all is this guy, okay? This one was discovered at 2,000 meters depth in Guaymas Basin. This is off the Gulf of California. 30,000, now this really blew my mind, okay? Because at this level of radiation, it should obliterate everything. But this organism not only survives, it thrives. It needs that. So because it needs that, you will have to constantly subject it to that level of radiation. The only place where I can do it is a nuclear reactor. That's why it's very difficult to grow. Okay. This is one of my future interests if I do get funding to do this kind of work. But the overall picture that you get is that the levels of extremity that biology can survive is keeping on pushing our limits of imagination and limits of reality. And what does this implies that we do not know for yet if life survives elsewhere. But the more we discover life and its extremities on planet Earth, the more it not only amazes us, we can use it as a template to look for life elsewhere, whether it is present or not. Okay? So to summarize, life has evolved to thrive in environments that are extreme only by our human standards. You can find them in the battery acid of Yellowstone, miles beneath the Earth's crust. There is another field called subsurface microbiology. Maybe I'll talk about in another talk, in another presentation. In pure salt crystals like halophiles, and inside the rocks of Antarctic dry valleys. Tomorrow, anytime in the future, we may send space shuttles to any of these places. When we do, all these analogs and the microbes living in them will serve as a template. For example, the pigments, right? We can use a like spectrophotometer, look for pigments similar to what we find in hypersaline to see, if we do find some spectral signature, maybe that could resemble life still present, or it was there in the past. We do not know yet. Okay, so this is not the end of, this may be the end of my current presentation, but this is going to be a never-ending field. And what you see here is a colorful moon of Jupiter, Io, which is also, I think, thought to have characterized by very, very violent weather phenomena. So as we enter to further ambitions is unknown, we do not know what surprises await us. Okay, so I'll finish this with a quote from Carl Sagan: "Somewhere, something incredible is waiting to be known." So astrobiology, right? It is science, but it is also a field of hope. We do not know if life exists, but the more we see the extremes of life on planet Earth, it sparks our imagination. Maybe there is something out there. I would like to acknowledge my parents, my extended family, all my teachers, including my great mentor, Dr. George Fox. George Fox, he recently retired. He's an emeritus professor. He's a co-founder of the Third Kingdom of Life, Archaea. All my friends and 2 residents of Estes Park, Stephanie and Sean Cosby, who saved my life on the 27th of May when I went on a hike. I should have, be gone that night, but they came to my rescue at 11,000 feet. So I'm that kind of an adventurous guy. Okay, last but not least, my favorite point. It's a take-home message. The woods are lovely, dark and deep, but I have promises to keep and miles to go before I sleep, and miles to go before I sleep. Robert Frost. And I thank you all for your attention.
That was cool. Well, I was wondering if anybody had any questions.
Thank you. Doctor, I was wondering if you have any opinions concerning Dr. Levin and his belief that he had discovered life on Mars through his experiments in the 1970s. They sent, obviously, several different probes to Mars, and Dr. Levin had designed experiments to actually conduct experiments on Mars. And as a result of those experiments, he concluded that he had found life on Mars, but almost all of the scientific community disagreed with him, but refused to repeat the experiments at any point later on when he begged them to do so, because that's a principle of scientific theory, that you repeat an experiment to demonstrate that it is or is not a valid one. So his experiment, which is the same kind of experiment that is used to test whether or not there are, active pollutants in water on Earth, he used the exact same experiment, sent that experiment to 2 different places on Mars. It showed signs of life, but the entire scientific community has rejected his conclusions. Yet they refuse to repeat the experiment. So I was wondering if you're familiar with Dr. Levin, if you're familiar with his theories, and if you have any opinions about them.
Oh, you're muted. It looks like on your end.
Oh, can you hear me now?
Yes.
Oh, yeah. So I've briefly heard about when I first came on my to do work on my planetary biology internship with Dr. Fox. Yes. Well, the jury is still out there, and the problem with I think the issue that people have is because of the extreme high levels of radiation found on the surface of Mars. I mean, I've not read his papers in detail, but if those were findings from the subsurface, then that would have been more interesting. But given what we have found on Earth so far, the level of radiation found on the surface of Mars, they'll be extremely detrimental to, because even the 30,000 grays, right? They are much lesser than what is found on Mars. I don't know the exact number, but I think that's the reason why they probably did not pursue it. But I need to get back on that because it's been a while since I read work related to his findings. This is to do the Viking, right? The Viking findings.
Yes.
I think so. Yeah. Yeah, but I'll tell you one thing. One of the prevailing, not one, I think there are at least two prevailing theories. The moon is definitely, it's a chunk of Earth, right? And there is a theory that in one of those interstellar collisions, something hit planet Earth and pieces of it might have fallen on Mars. If that is the case, right? Even if life has become extinct, there has to be signatures of Earth-like biology, not life, Earth-like biology there. We do not know. So we need many more such probes that are sent to Mars with more spectral analysis instruments, which are advancing by the day. And there is only a finite number of missions we can send to Mars, unless of course you have many more people like Elon Musk come into the picture, but that presents a different set of problems. So personally, if you ask me, the problem is if we are looking, it's a conundrum. It's a dilemma, if you will. If we were to look for Earth-like template, use Earth-like biology as a template, we may actually miss something that is non-Earth-like. Am I making sense? Like if you're using DNA, RNA as a template material, all the spectra here, if there is something that is highly unusual, non-Earth-like, we will completely miss them. But then we don't have anything else to start with. So those are the problems. I do not know if I have addressed your question in entirety, but I'll definitely give my contact with Bill. Reach out to me. I will do more research and get back to you on this.
Awesome. Does anybody else have any other questions?
So there are a number of moons, planetary moons, in the solar system that are thought to contain water. If you had your choice, where would you send a probe to look for?
Are you referring to things like Titan in Europa?
Yes.
Yeah. I would love to send one to each of them. I'm very greedy.
Well, I looked it up on ChatGPT and it said there are 5 moons. Triton, Titan, Europa, Ganymede, and
In fact, I would love to do another presentation someday because I made this presentation a few years ago in Switzerland. I think Ganymede is the moon that gives out this springs of water, jets of water, right? That reach a very great height. And what is interesting, these are actually water. And the idea is that I'm really curious to send something to that place because if over 30 years of adaptation or exposure to stringent conditions in the spacecraft clean room can make the spores increasingly resistant to higher levels of radiation. Okay? If something seeded something there, okay, which may initially not have survived, something survived in the subsurface in the planet, on the planetary body, not planet, moon. Over periods of exposure and adaptation, they might have gotten adapted to. In fact, there is somebody I don't want to misquote somebody's name, but I think it could be Kevin Han. I think they are actually doing these kind of experiments. It's called directed evolution, to see you take an Earth microorganism that is already resistant and keep on exposing it to incremental levels of extreme conditions to see how far they can keep on adapting and evolving. So, but you know, I'm digressing. To answer your question, I would love to send one to everything
It sounds
if
like
there was enough money.
sounds like a great theme for another movie, directed evolution. Yeah.
Yeah.
Thank you.
I didn't cover some of the directed evolution work that we have done because it'll, as I said, 50 minutes is for me 50 minutes sometimes in one meeting. Okay, since I told you about how I did astrobiology outreach, right? I didn't even know about something like this existed. In the year 2012, my boss, George Fox, he received an email from NASA. They were organizing a competition called FAME Lab, FAME Lab, which is like you take established scientists or postdocs, they have to speak about science for 3 minutes, addressing a public audience. And it has to be relatable. You can do all kinds of goofy things to speak about science, but you have to convey it without dumbing down the science. I got annoyed. I said, I'm supposed to be doing research, not doing these kind of goofy things. I'm not Sheldon, from that series. But there was nobody to represent our lab, so I did. And I realized that I actually enjoyed it. So what I didn't want to do, I began doing it, and then it became like a habit. So in the very next year, Death Valley National Park, somebody who was in the FAME lab invited me. Death Valley National Park, along with the National Park Service and NASA and other NASA agencies, NASA Ames Center, they organized a festival called the Mars Festival in the Death Valley National Park. That was the first time where you had like a community of scientists there giving goofy 1-hour presentations to the public. You have to explain science to members of the visitors there, the visitors to the park, who are coming from all over the country, even the rest of a lot of people from Germany. You have to give them a presentation in an auditorium, something about this work. This kind of outreach began there. So since then I've been doing this, and definitely I would love to do a few more, at least one more. I didn't cover many of the things that I actually do because as I said, 50 minutes is just like an introduction for me. But I'm hoping that it was interesting enough without going deeper into the details. And one thing I've realized is in many of my presentations, the more if I go deep into the technicalities, people start losing interest and they falling asleep. So one thing at a time. I cannot take an entire semester's classes in 1 hour.
It's a giant topic.
Absolutely.
Doctor, thank you so much. We really appreciate it and hope to see you again.
Yeah, it is my pleasure. How come I don't see the others? How come my screen is still small? It says I'm screen sharing. Am I still screen sharing? All right.
We still see you.
Okay, you see me, but you don't see my screen? Okay.
Sure.
Okay, how do I enlarge my... Wait. A participant has enabled closed captioning. Oh, okay. Oh, I see some... Oh, sorry, sir, I didn't see the chat at all. Okay, Michael asked that question, okay. My question surrounds the potential for life in liquid methane on moons like Titan. I will do one thing. There is an expert on Titan that I know of, okay? I think she's at Cornell. I will see if I can persuade her to come and give a talk to you guys, or I'll give a talk on Titan. How about that?
Deal.
Yeah, not just Titan, Europa, and Ganymede.
All right, well, thank you so much. Robert?
Yeah. So I can mute myself now?
Thank you very much, Doctor. I think it was an outstanding presentation. And let's all give it up for him. Thank you very much. So let's take a quick break and then or hit our raffle and get on to another key presentation, what's up in the sky in the next month. But 10 minutes?
That's gonna.
No, I'm having a little trouble getting. What if you had any better luck?
I did. 0640. I can't be that boring, Matt. Yeah, 2950640. Yep. Okay. 2950661. Come pick up your prize. 2950661. We can't have duplicates, can we? That matches that. 2950661. Okay. Okay. Yeah, I didn't want to necessarily be arbiter. Nothing. 2950602. 2950602. All right, we have a winner. Well, we have 3 winners. Donated by Arizona. Yeah. Okay, and now we're going to have Bill give us his monthly presentation about what's in the sky, if El Nino will let us have a look.
It's just insult to injury, isn't it? So, what is in the sky? This is for the next several minutes, I'm going to give you hope that there is something out there beyond what we've been seeing for the last month. By the way, this is a local reflection nebula by our own Cole James. It is a water molecular cloud, dihydrogen monoxide. So, this is what we hope to see in the next month. Next slide. Okay, so we've heard of galaxy season and we've heard of ISS season. Well, this is Jovian double transit season. So just every once in a while, and it doesn't happen a whole lot, that the moons of Jupiter kind of get in that sync right there, where we get these double shadow incidents that happen pretty regularly. This is actually happening for the next few weeks. Wednesday, tomorrow morning, 7:17 a.m. It's actually kind of a cool start to this. It is a moon occultation of Jupiter. And when the moon passes from in front of Jupiter, or you want to say when Jupiter shows up on the other side of the moon, it's going to have 2 shadows on it. So it's kind of like this extra little bonus thing. Next slide. And this is the schedule. So you're going to have to fact check me on this if you're interested in these sort of things. If you've never seen one before, it's really cool. It's just neat because it's, you know, you're seeing some depth of field with the moon relative to Jupiter, and some of these aren't exactly at opportune times, and some of them are. I'm not even sure if I can verify if every one of these is actually visible or Jupiter's even in the sky. So that's kind of where the fact-checking part comes in. But this is the schedule. So happy double transit season. So, of course, I'm not even going to say, you know, given that the weather is allowing for this. So next slide. All right, well, the next new moon is Friday the 19th. Next slide. So we've got Mercury in the morning, and that's on May 29th. We're at greatest western elongation. It'll be about 14 degrees up at its highest from the 30th of May to the 8th of June. It's always a neat sight to say, hey, okay, I've seen Mercury now. Next slide. All right, Venus and Mars and the beehive. So, you know, for all of you that are interested in conjunctions and stuff, Mars actually makes a pass through the beehive cluster. So it's a nice visual and maybe photographic opportunity on June 1st and 2nd. And Venus does it as well on June 12th and 13th. So this is actually the beehive cluster right here by our very own Christopher Ashford. So next slide, please. Yeah. So full moon's on June 4th. Believe it or not, see it or not. Next slide. We got the next new moon on June 18th. Next slide. And the constellation we're going to talk about this time is Hercules, the hero, one of the 2 heroes in the sky. Can somebody tell me who the other hero is? There you go, my man. So Hercules is it's an old constellation. Hercules, the name is actually Roman. The Greeks is Heracles. The Sumerian called it Gilgamesh. And it's got 12 stars with known planets. It's got 11 named stars according to the IAU, all of which I've never heard of in all of my experience with amateur astronomy. But there they are. It's got 2 Messier objects. It's got a prominent keystone asterism, and well, what he's wearing there is it's Nemea, which is Leo in I think Mesopotamia, and basically he killed the lion and put it on to make him even more creepy than he already is by offering bouquets of pomegranates and waiting to bludgeon you or something. I'm not really sure what Johann Baer was thinking when he draw this, but you know, that's what's going on here. So next slide. Okay, so the Tau Herculids. So we kind of heard about the Tau Herculids probably the first time last year. Made a little splash in the news thinking that it was going to be maybe a major meteor shower. It did not turn out to be the major meteor shower that we all hoped for. June 9th is its peak, And its material source is 73P Schwarzmann-Wachmann. And it's actually a star, the Tau Herculid is actually one of the stars that makes it around to be our northern celestial pole star in 18,400 AD. So, save the date. Next slide. And then there's HD 147506. So I don't put these on here because you're like, hey, this is going to be something you might see. This is so you know what's going on. I could tell you all about the big bright things, which I do, but I also tell you about these little weird things that are interesting facts that you may not know and may never hear about with these constellations that I cover. So it's actually known for having the most massive transiting extrasolar planet. It's blah, 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 the name of the planet. It's 9 times as massive as Jupiter, and it orbits the star every 5.6 days. So we're talking about a super hot super Jupiter. Next slide. So then there's Messier 13. I think we're all familiar with it. Big, beautiful globular cluster right there in the keystone. Some say that there's up to 1 million stars. I've heard numbers anywhere from 300,000 to 1 million. The Arecibo message. So this is a fun fact about M13. Next slide, by the way. By the way, this is a picture that our David Cox took of that with a beautiful galaxy. I don't know what that galaxy is up in the corner there, but really nice picture of the globular. But the Arecibo message was sent in 1974, and the whole idea was, well, we're going to send a radio message out there, and we're going to point it towards M13 because M13's pretty dense with stars. So we're thinking, okay, the likelihood that there's a planet circling around one of those stars that has life. But they didn't really think it out because by the time the message actually gets to M13 is going to be in a different part of the sky. So, they didn't really think that out, but there's your fun fact about M13. Next slide. So Messier 92 is the other Messier object in the constellation. It's actually about the age of the universe itself. And it was actually discovered by Johann Bode. Next slide, by the way. It was actually discovered by Johann Bode 4 years before Messier did, and cataloged it. This picture is actually by Christopher Ashford. And next slide. So, Abell 39. So I'm a big fan of planetary nebulae. The Bell catalog is actually really, really faint planetary nebula. Really requires a dark sky. Next slide. The cool thing about this one, this is what I love about these things. Of course, planetary nebula are emissions of a dying star in the middle. And this one's actually got a radius of about 2.5 light years. And it's really close to spherical. And it's actually one of the largest known of nebulas of this shape. Just a beautiful bubble in the sky. Next slide. So then there's the Hercules galaxy cluster. So this has got about 200 galaxies in it, and it's actually pretty dense with spiral galaxies. So the address, if you go to the next slide here, there's our cluster right there. The address is Hercules Galaxy Cluster. That's in the Hercules supercluster, which is a part of the Great Wall, which is another structure that is claimed to be one of the largest, if not the largest structure in the universe. Next slide. So within the Hercules galaxy cluster is an ARP object. And I love ARP objects because safely from far away, we're watching destruction on an enormous scale, a galactic scale. Next slide. This is actually a Hubble shot, and it's got these two galaxies, got in GC 6050 and a index catalog 1179 that are in collision. When I first saw this picture, I thought, well, now, hold on a second. I see 3 cores. What's up with that third core up there at the top? Maybe it's a satellite galaxy. Sure enough, that's what it was. It's that's the name of the galaxy right there. A bunch of letters and numbers, but it's just a smaller galaxy that just got caught up in the mix. But just a beautiful interaction of disaster going on out there in the Hercules galaxy cluster. Next slide. So then there's Hercules A. So it's a galaxy in the middle of it called 3C348, and it seems like a regular elliptical galaxy when you image it and look at it in visible light. But what they did was, thank you, they imaged it in radio waves as well. And these are plasma jets that are a million light years out from the galaxy. These beautiful plasma jets coming from the central black hole. And you wouldn't know it unless you were imaging radio waves. So it's just a spectacular sight to see. I wish it was something we could see in person. Next slide. So here are the websites here. These are the ones that I always reference, theskylive.com and constellation-guide.com. It really is a great resource for a deep dive on objects that are going in constellations, not just deep sky objects, but the stars as well. And they'll talk about what's special about the stars. Sometimes you come across some really cool stuff that's going on out there that's more than just being a star. So next slide. So, like I always keep telling you, keep looking up. And when you can get a break in the clouds, squeeze it in where you can. Thanks.
Thanks, Bill. It's always a great presentation, and written down some things I'm going to try and image in the next month. So again, thanks a lot, Bill. So we're going to jump right into monthly reports. Patrick? You're going to have to come down here, buddy.
All right, well, I got 2 star parties this week that will most likely be canceled since we can't seem to get rid of clouds. This Friday, we have one for a Cub Scout group in Keller. This is our 3rd attempt, probably a 4th attempt. We're going to try to get this one in. And then Saturday is our monthly Dinosaur Valley State Park, but the forecast isn't looking good. If that clouds out, do have a backup date of the 27th. I'll let you know if that happens. That's all I got.
Thanks, Patrick.
Hello, everybody. I'm Bruce Campbell, and I'm representing Michelle for support from the development. So we just want to give you an update on where the video, where we've been going. So in 2020, we fell in love with
2022.
2022, thank you, fell in love with this property. And unlike the other dark sky sites that we'd visited and had used, we had no we had no access to. This really was just a parcel of land, be a pasture. There are no pathways, observing field, buildings on site, as it says. Fences didn't necessarily have fences all the way around, and we had a field that had cactus, trees, rods, and holes. Really a mess in terms of an observer. But since over the last 10 months, 11 months, let's go to the next slide, the development committee has been busy transitioning this pasture land in. We now have a building on site for our composting commode, so restroom essentially, and we do have telescopes and stored in there. They're available to anybody that goes out to visit. We constructed a private entrance to the county road that we use so we can actually access the property, and we're no longer relying on good graces of our neighbor that allowed us to. We've cleared a pathway through the trees so we can actually drive on our property to the observing field, and we worked hard over the last 6 months to remove most of the cactus and tea trees, although we've heard some experts in the area that if we didn't get the roots out, we have that same problem. So we'll be working on that in the coming year. We leveled most of the observing field so you won't fall in any ruts while you're out there, especially in the dark. Good thing. And then the solar panels have been brought back into service. We also installed red lights using power that were from the solar panels to illuminate the inside of the green. So we've accomplished all these tasks, but we have a few more things to do to really finish phase 1, getting this property the observation. We want to install a barbed wire fence on the north and east property lines. We want to install a culvert at our access on County Road 244, and then we need to install or build a storage building. And because of the cost of all these, the development committee has got to go to the executive board for approval, and once the plan is approved, we'll come back to the membership, explain what we plan to do, how much we plan to spend, so you know what the costs are. So I'm giving you an insight of what's about to come out of the development.
Thank you, Bruce. Tom, do you have anything on Palo Pinto?
So as of date, we've had out of 115 members, 42 on the partners board have volunteered to clear 3.2 miles of trails. We're taking care to create outcroppings to look at the scenery and stuff. We have 9 more miles to clear out. We're suspended from this work until the fall because of some nesting birds that are taking up residence there, not allowed to work there. Partners board meeting is May 27th. Janice Janes, that is in charge of that, has been taking classes on grant writing. So we're thinking long-term as far as different programs of the state park, including astronomy. We're looking at some better spots to put at the astronomy site. Then hopefully we'll be involved with some development, making that a go-to place for astronomy as well as rising stars. think long-term, they project 80,000 visitors a year at the state park, and I look at it as a feeder system to increase our membership. So we're putting some sweat equity in the beginning, and it's a very driven partners board to make this park really good, and I think very optimistic about what will do for our club in long term. So
Thank you, Tom. Mr. Legal Beagle. And we should be jumping for joy, and I'll tell you why. How often does a lawyer bring you good news?
Good news on two fronts. First, we got the property tax exemption, so we don't have to pay property tax. So that's going to save us about $1,500, which would be equivalent of 30 memberships. So that's very good. Anyway. All right, there we go. Number two, sales tax exemption. We got that all fixed up. So if you go to Groups.io and you go down here to files, I'm going to stand over here. Go to files, go to the third page. You come up here, Texas tax exemption form. Next slide. You'll see three things. There's some archives. Our exemption certificate, the database snapshot, and the sales exemption letter. The last one's the most important. Next slide. If you go to the database, you just put in our magic number and it'll say what we got. If you hit that other thing where it says view letter, next slide. This is the letter that actually says we have the sales tax exemption, plus a couple other things. So if you're buying stuff for the club, have a copy of this. Next slide. This is the most important part, the sales tax exemption form that we have to submit. You'll see it has our address. Right here it has our information on our charity. So things you got to fill out up on the top right, phone number, that stuff in the middle, seller, yada, yada. I'm buying a bunch of stuff, how much it costs, and sign it, dated on the bottom. So if you're buying stuff for the club, please use this. It'll save us some money. So we can use it for these things, probably our websites, all the rest of that stuff. If you're buying some equipment, so it'll help save us a couple $1,000 a year. Depends on what we're buying. It'll be great. One caveat, don't use it for personal business. Three things can happen there. One, you can be prosecuted for fraud. Number two, we could lose our status. Number three, and most importantly, me and Phil and a bunch of Marine friends will come to your place in the middle of the night and do a little code red on you. So that's all I got.
Thank you, Cy. We've been working over the last year to get some things organized, and a lot of the legal stuff that happens behind the scenes, Cy does for us. When we ask him to do something, it's done quickly. So, and a lot of this stuff is, it's confusing to me, that's for sure. So, just a few things here. Actually, my list is long. It's really not that long. Someone asked for us to put the documents on the purchase up on the e-group. So Cy is definitely working on that. He has all the paperwork. He's going through it to make sure to redact any PII, which there is some. Then we'll get that posted. It's not something that we've forgotten about. So next month is election month. And we have one open director slot. Our John Giarmini, who's been our treasurer for I don't know how long, is not going to run. So we are in desperate need of someone to stand for treasurer. John is going to be around to partner, and in the IT world, we call it KT, knowledge transfer and all that stuff on what the treasurer does, not the John Giarmini treasurer. So he's definitely going to be around. He's also going to, as I mentioned earlier, he's going to help get memberships squared away. And I was just told, how many? 197, we grew by 2. So we're getting there. So in order to vote, if you haven't paid up your membership, you need to do that. And you need to be present. We need to be present here at the meeting or online. There's no mail-in ballots. Okay? If you're interested in any of the positions, let us know. The board as it is now, with the exception of John, and we do appreciate his service tremendously, will be standing for reelection. Yep. So any questions on elections? Chris? Okay. Some really exciting news, thanks to Bruce. 469 is the magic number. The address of the Rising Star Observing Area is 469 County Road 244. Rising Star, Texas 76471. Okay. And Bruce put a little sign under the our signage, and it's on the left there. We don't have a mailbox, and if we can get away with it, we won't have one out there. But we have to find out, there may be some postal regulation or something. Now that we have an address, then the post office may want to know what to do with anything that shows up there. So anyway, so that's exciting news. So when, if you're out there and you have a problem and you call 911, you can give them the address and they'll come on out. So, and hopefully if you're out there, there's at least 2 people. So here's, it's funny and not funny. So we have 4-legged members. They're called cows. And they do make appearances. They jump over the fence. And I mean, you wouldn't think they could, but they do. We don't have any holes in our fence right now, but they do jump over. So keep in mind, the cows are much bigger than you. Don't chase them. Don't try to spook them off. They're not going to bother you. And if you're out there and cows show up on the property, go ahead and give me a call. I will contact the neighbors. I don't care if it's 2 o'clock in the morning or 7 o'clock in the afternoon or evening. Just call me and I'll call the neighbors. They are responsible for their animal. Okay, so but don't chase them, because that's just asking for all kinds of. And also,
I have a question about that.
yeah.
They're aware of the issue, right?
Oh yeah.
Have they been?
Oh yeah.
Okay.
Yes. Did I say yes? I've
Yeah.
spoken to them. Yeah, just they generally cattle, bovine are very timid. But if you spook them, I mean, they're like any other critter. You know. Let's see. And lastly, we are going to, on June the 8th, we're going to be doing a fairly hefty solar event for the HEB school district. That's going to require us to be at 4 elementary schools in the HEB district. Chris Malicki is going to help us do EAA on the day, but there's some things that we're going to need help from the solar guys. So we will be reaching out to you, okay? On June the 18th, which is a Sunday, and a full moon, or not a full moon, a new moon, HEB School District has asked us to host a star party for them at Pennington Field. It's going to be pretty big. So I should have more details probably by Monday, and I'll get a blast out. We're going to need quite a few folks out there. So I think it will rival Dino Valley last year on their anniversary. There was something else, but I can't really remember it.
John Reese? Someone
Oh,
named
John.
Reese? Is it
John
you?
Reese has joined us. He's got some equipment he'd like to talk about.
Yeah, got your mic here.
Thank you. Appreciate it. My name's John. I've been a member for about 6, 7 months. This is a bit of a show and tell and sell. We had recently a member of the Fort Worth community contact us about a telescope that was his father's. His father passed away, and he thought to contact us because he figured we might have a good use for it, or someone here might be able to good use. Yeah, appreciate it. It's a Celestron Super C8 Plus. It comes with everything. It comes with the tripod. The rest of it is out in the car. It has a 2,000 millimeter focal length. It's pretty nice. I've actually tested it out. The optics are really clean. It comes with a bunch of eyepieces and accessories. And it has a buyer's drive motor that powers up, lights up. It spins, the worm gear spins, but the telescope doesn't spin. Clutch has to be that. And he has very generously agreed that he's asking $750 for the whole thing, all the parts and accessories, books that come along with it as well. And he's give half of. If interested, I have his information here. I don't know if you wanted to say anything else or. So down here, there's a list. Oh, sorry. There's a list of items, including bang, plus of accessories.
Thank you. There was one other thing I wanted to mention. We have 2 solar eclipses coming up in the next year, and Chris Malicki and Juan Martinez have volunteered to facilitate the club's activities with regards to those 2 important events. So I wanted to get that out there. I'm sure we'll be hearing from them. They have a lot of great ideas and plans. So if you have any thoughts or questions, you can contact them. And again, we'll be hearing from them as things move on. Now for the fun part, we get to hear from our august treasurer and get our financial report.
I thought I'd come up to the front. Nobody ever gets to see me. You can read the numbers. I'm not going to talk about that much. We are getting rid of our savings account because
because there's no money in it and we have to carry pretty substantial amount to avoid the fees. So the bottom line is this $15 fee took all the interest that we earned on that account in 5 years. Because you see it was paying 0.001 APR. So anyway, that's a work in progress. Actually, Robert and I both have to go there. What we have upcoming major expenses is the payment of this room from July forward and the Astronomical League memberships, which is due June 1st. So this number is out of date by 2 hours. We're now at 197. We had 2 waiting, 1 fell off, 2 took their place. So there's a possibility that in 2 weeks we can make it 200. That would be pretty impressive. Next. And so that's where this graph is. I've never seen the graph cross the 200 line. Be nice to see that. Go ahead, next. And this is where we are on renewals right now. So go on up some, oh, right there. And know some more. Yeah. So we had 177 people that are paying customers. Of those 177, we've had 62 people plus, I don't know, 15 or so today that have paid. 3 bills that came in while we were in the meeting. So we're probably up to 80-ish, thereabouts, but 80 is not a lot when you compare it to 177. So that's what we're working with. And just so people know, these are the ways we collect money. And checks were from 17 people, and 5 people use Zelle. So majority of folks are doing that. Go ahead. One of the more interesting things that happened during the last month is we got a request from a person in a school in Euless to assist a student, a 5th grader, I think it was, with 4 questions that this person had. And I actually went out of my way to really give them a term paper on the questions she asked. But the teacher never got back to me. I was so upset. I called the place down there and got her number, left her a voicemail. I don't know, maybe she didn't like the moon shot with the plane coming out of it. So we've sent the second email. There's 3 emails in the series for renewal. If you didn't receive the second one, that's because you paid already. If you didn't pay, that's why you get the email. There will be 3, so we'll send the final one the first week of June. And I got something to play with. Thermal printer. My intent is to not have to write receipts anymore. So I'll just pick your name out of the roll, take the money you're designating, and just print the receipt for you. But I didn't have time to get all that work. Came in yesterday. Any questions for me? I got some interesting questions when I was at the bank today where they were asking me all these things, and one of them was, do we take cryptocurrency and Bitcoin? And yes, well, no. Yes, Tom. That information is, I don't know that I put it in there for 2022. It is available. I don't think I've actually put it on e-groups yet.
but I've done the pivot table and the graphs and all that wonderful stuff. I think we're 4,000 ahead of where we were last year in the checking account. Of course, the other one doesn't really matter. We know where that money went. There's also on in the monthly financial report for May of this year, there's 3 PDF files that outline the expenses for what's been spent at Rising Star. So if you ever wanted to know, there you have it. Answer your question, Tom? Yes. Stepped in what? Renewal. 60 for family, 50 for individual, 30 for students. That's oh, I was trying to keep it clean, so. Anybody else? Anybody from online ask a question? No? Good. Super. Thank you.
And by the way, in the hopes of keeping John as treasurer, I gave him a raise from 10 zeros to 15 zeros before the decimal point, and that didn't work. There's nothing on the infrastructure or marketing report to talk about other than we're starting to build a plan for marketing what we do and asking for grants to help pay for the things that we need to accomplish and do out at Rising Star. There's nothing to vote on today. Anybody have any questions or comments? The floor is open. Oh, I did have one other thing, but I'll wait till we're finished. Yes, sir. Yes, Matt. Yeah, that's pretty good. Hey, show of hands, how many people have been to 3RF? Maybe I should ask this question. How many people have not been to 3RF? Okay. I went 2 weeks ago. It is an awesome place. If you haven't gone, you got to go. Jerry Gardner, who's a contributor of the club, is the astronomy manager out there. And it's an easy 3-hour drive from here. So if you haven't gone, you should think about it. And just a sec. And for the Astro guys, he's going to host a 4-day Pix and Site conference in December, starting the 7th. It's a Thursday. I've already signed up. Okay, I'm sorry. Go ahead. I was going to bring that up next month, but so the short answer is we're starting that process. The real question is, you know, when and all that kind of stuff, and what the requirement's going to be. I was talking to Cy about that. We need to, we can go out there pretty much when we want. but we need to get a formalized agreement with them so we understand what we're going to do and they understand what we just have to have a agreement with them, and that's something that's in process. The Thompson Observatory is it's a Bortle 4. It's about 40, not even 40 minutes, it's probably 30 minutes from here. It's in Keene, Texas. It's part of the Southwest Adventist University. It's a pretty cool little place. Any other questions or thoughts? Make a motion to adjourn the meeting? All in favor? Thanks a lot, guys.